Alright guys, so today's notes we are going to cover some of the cell processes, more specifically the movement of things across the cell membrane. So the previous notes we went over the major cell organelles, and today we are going to focus on the cell membrane. Alright, so the cell membrane was that, little bubble that kind of holds the cell together, and it controls what goes in and out. Okay, so one of the terms that we use to describe the cell membrane is that it is selectively permeable. Okay, so basically it regulates movements of stuff in and out of the cell. Alright, so this cell membrane has very unique characteristics. Okay, so I'm going to do like a really rough sketch of it, a zoomed in version of the cell membrane. Okay, so the cell membrane is made of these kind of funny looking structures, and these are known as phospholipids. Phospholipids. Okay, so this would be one phospholipid, and because it's made of two, we say that the membrane is a phospholipid bilayer because it has two. Okay, so some unique characteristics about the phospholipid is that these little tails are going to be hydrophobic. Okay, and if you think about the word phobic is scared, so hydro means water, so they basically repel, they hate water. Okay, and then the head is going to be hydrophilic. Okay, hydro, water, philic is loving, so likes water. Okay, so this is the tail, and then this is the head. Okay, so if you look, the heads are both towards the outer part of the membrane. So this side would be the extracellular space, so outside of the cell, and in here would be the intracellular space, so inside of the cell, or also known as a cytoplasm. Alright, so this phospholipid bilayer is going to play a big role in how the cell controls what goes in and out. Okay, so some kind of basic tips, I guess, would be if it's really big, so things that are big or charged can't just cross through the cell. Can't automatically cross through. Okay, but if they are small and not charged, so small, lipids, maybe some steroids or things like that, and not charged, can usually just cut through, just cross through the cell. Alright, okay, so there's a couple different forms of transport, okay, and they can pretty much be categorized into two sections, or two main categories. So transport, moving things in and out of the cell. So these are the methods by which things cross the cell. Okay. Like I said, there's two categories. Okay. The first category is known as passive transport. Okay. So passive transport is going to not require energy. So no energy needed. Okay. This is going to move towards equilibrium. And we'll go over that in a little bit. Okay. It wants to balance everything out. Okay. So this is going to go from high concentration to low. Okay, because it wants to reach that balance, that equilibrium. Okay, and we say that this goes with concentration gradient. Okay. And this is going to subdivide into three types. Okay. So type 1 or type A is going to be known as simple diffusion. Okay. You also have facilitated, kind of like the name says. They have a little bit of help, but still no energy. So facilitated diffusion. Okay, and this is going to use things like channel protein or channels or transport proteins. And then the third type of passive transport is going to be osmosis. And osmosis is just only deals with the movement of water. All right. So, if you look at the handout for the notes, okay, we are going to kind of just show this with some images to kind of help make sense of this information. Okay, so I'm just going to stick this in here. All right. Okay, so the big thing here is that you have things moving from high concentration to low concentration. Okay, so this one is just, the molecules are just moving right through the membrane. They don't need any help. So this would let you know that this would be diffusion. Okay, these two scenarios over here, you have things also moving from high concentration to low concentration. So they're moving with the concentration gradient, okay, but they need a little bit of help. So this would be an example of a channel, and this would be like a transport protein, okay. So because they need a little help, this is known as facilitated diffusion. Okay. Osmosis would look pretty similar to you know, either one of these, except you would know that the molecule that's moving is water. Okay, so these show passive transport. Okay, so you have your cell membrane. Okay, you have your extracellular space. And this would be your cytoplasm. Okay. And stuff is moving into the cell without the use of energy. All right, the next type, or the second category, so this is category number 2, this is going to be active transport. All right, so active transport, the big giveaway for this is that it needs energy. Okay, and the reason it needs energy is because you're going to put stuff, or you're going to be moving stuff against its concentration gradient, meaning you're going to be moving things from low concentration to high concentration. Okay, things naturally want to kind of level out, so you have to go that extra step to move things against their concentration gradient. Kind of like if you're swimming with the current, you know, you can kind of just float by, but if you are trying to swim against the current, you have to put that extra oomph, that energy, to move in that opposite direction. Okay, so you would say that this goes against concentration gradient. All right, so under the second category of active transport, there's going to be two types of active transport. Okay, the first one is going to be cell membrane pumps. So you're going to have protein pumps embedded in the cell membrane that are going to be pumping stuff into the cell or out of the cell against that concentration gradient. Okay, and then that second type is going, so this is type A, and this would be type B. This is going to be the movement of vesicles. Okay, and so you have a subtype. You have things that fall under endocytosis. And if you look at this word, endo means in, so it's going to go into, cyto is cell, so into the cell. And the two are going to be pinocytosis. Okay, and this is going to be for small things, so for example, solutes or small nutrients. And then the other type is going to be phagocytosis. And this is going to be for big things. For example, like if it's a white blood cell that's engulfing or eating a bacteria, you would say that that is phagocytosis. It is eating an entire cell. All right, and then the other type of a movement of a vesicle, so this is subtype 2, is going to be in the opposite direction, which is going to be exo, which is out, cytosis, which is out of cell. Okay. All right, so just like the other one, we are going to show kind of what this looks like. Okay, so this is going to be the other part of the handout. Okay, so the big giveaway for this image here, where you have your cell membrane, is that you have this protein, but you require energy. So that's the big giveaway to distinguish between passive and active. Okay, so this is going to be active transport. Okay. Which is also, you know, this entire category is active transport. Okay, if you wanted to be a little bit more specific, you would say that this is active transport through pumps. Okay. All right. Okay, so these last three images are the movement of vesicles. So you kind of just have to pay attention to the direction that the things are moving in and how big the stuff is. Okay, so this one, you have a pretty large thing, okay, so we're going to say, let's say this is a bacteria that is being brought into the cell. So this would be a form of endocytosis. And more specifically, because it is a large thing, this is going to be phagocytosis. Okay. This one is also bringing stuff into the cell, but these are small solutes, so this would be pinocytosis. Okay, so both of these are an example of endocytosis. Okay, and then this one is being very clear that the contents of this vesicle are getting kind of dumped out of the cell. So this is going out of the cell. So this would be exocytosis. Okay, the very last thing that I wanted to go over is just a really quick recap of mitosis. Okay, so hopefully you remember mitosis a little bit, IPMAT, and if you are a little fuzzy on it, I would do a rough sketch of the pictures that I have here on the screen. Okay, so just a little review. The first part of the cell cycle is going to be known as interphase, and interphase, you can really tell it apart because when you look in the nucleus, the DNA looks just like a jumbled pile of noodles. Okay, you should still be able to see the centrioles kind of just clumped together somewhere in the cell. Okay, when the cell is ready to go into mitosis, okay, and you enter the prophase stage, you're going to see that the chromosomes have now condensed. So you have your copies made, your centrioles kind of begin moving to opposite sides of the cell. And then the metaphase, we're just going to kind of, this is the in-between, but the next big noticeable stage is going to be metaphase. So meta for middle, you're going to have the chromosomes lined up in the middle of the cell. And then anaphase, A for the chromosomes being pulled away or apart. So the spindles, help move the chromosomes to either side of the cell so that both cells can have a complete set of chromosomes. Okay? And then that brings us to the telophase. Okay, so telophase, the way that you can really tell it apart is because you start seeing the formation of the two nuclei. So you see these little fragments here. Okay, and then cytokinesis refers to this kind of cut in the middle. That's going to give you the two cells. So cytokinesis, cell breaking, I guess. I like to think about it as kind of looking like a butt. Okay, this part here in the middle is known as the cleavage furrow. Okay, so cleavage, kind of like you see in some people. So anyway, this is a really quick recap of mitosis. So make sure that you can distinguish what is happening in each one of the major stages. So interphase, prophase, metaphase for middle, anaphase for being pulled away or apart, telophase for two nuclei, and cytokinesis is the actual cutting of that cytoplasm of that cell so that you end up with two identical cells. Okay, so that's the big thing for mitosis. Mitosis gives you identical, or I guess same. Alright, so like always, if you have any questions about anything that we have gone over today, just let me know and I will be more than happy to help you.